Okay, now that we have talked about the basic composition of the plasma membrane, we can start talking about the transport of molecules across that membrane. And the first important idea is that there are some molecules that can freely diffuse through the plasma membrane. Those molecules belong to either of three groups. The first one is gases, such as CO2 and O2. The second one is small hydrophobic molecules, such as benzene. And the last is small polar molecules, such as water and ethanol. Yes, this implies that the moment that you consume ethanol, the ethanol will freely diffuse through your plasma membrane and will rapidly enter the cell and exert all the different effects that ethanol can actually exert within the cell in no time. The second important idea is that there are specific molecules that, even though they are relatively small in size, cannot cross the plasma membrane simply by diffusion, passive diffusion. Those molecules require some sort of transport, and the transport in this particular case is given by carrier proteins or channel proteins that allow these molecules to enter the cell. And examples of these type of molecules that cannot enter the cell by passive diffusion are glucose and all large polar molecules, amino acids, and charged molecules such as ions. These molecules enter the cell by facilitated diffusion, which requires the presence of either carrier proteins or channel proteins. To better understand the process of facilitated diffusion, let's first take a look at those molecules that enter the cell thanks to the action of carrier proteins. The glucose transporter exemplifies facilitated diffusion by carrier proteins. So let's take a quick look at how it works. The glucose transporter is an integral membrane protein that contains a small cavity that is normally exposed to the extracellular environment. That cavity allows for the lodging of glucose into the center of the protein, and once glucose lodges in that particular place in the protein, it triggers a conformational change on the protein, which then leads to the opening of the cavity toward the intracellular side of the protein. The lower concentration of glucose that is present in the intracellular space of the cell leads to the dislodging of glucose from this membrane transporter, therefore allowing glucose to be delivered into the cell. The cycle then repeats itself. Once glucose has gone into the cell, the protein undergoes another conformational change that now allows for the opening in the protein to be exposed to the cytosolic side once again, therefore allowing for new glucose molecules to enter the cavity and once again trigger the conformational change in this transporter. Two important characteristics to keep in mind are the following. First, glucose molecules are moving in the direction determined by the relative concentration inside versus outside of the cell. Second, there are no energy requirements for the transport of glucose from the outside to the inside of the cell. Glucose is simply moving in favor of its concentration gradient. Besides glucose, there are other molecules that are carried into the cell by facilitated diffusion using a similar mechanism. Among them, there are other carbohydrates, amino acids, and nucleosides that use other transporters to enter the cell. From the molecular point of view, it is important to keep in mind that the glucose transporter contains 12 different transmembrane domains. And one important peculiarity of these transmembrane domains is that in their sequence, some of these transmembrane domains contain polar amino acids, represented in the figure by the violet balls. Such polar amino acids are usually absent in most transmembrane domains. However, for the glucose transporter, these polar amino acids are the ones that allow the protein to interact with glucose and serve as a transporter for this sugar. Let's take a look now at facilitated diffusion by channel proteins. Facilitated diffusion through ion channels is extremely rapid. It can be 1,000 times faster than facilitated diffusion through carrier proteins. Ion channels can be extremely selective, very highly selective. And the opening and closing of these ion channels is regulated by gates. This implies that those channels are not permanently open. Instead, they actually open only transiently in response to stimuli. Based upon the type of stimulus that is used to open an ion channels can be classified into two different types: voltage-gated ion channels and ligand-gated ion channels. The sodium and potassium ion channels exemplify voltage-gated ion channels. The selectivity of the sodium channel is dictated by the size, the inner diameter of the sodium channel. Sodium ions are hydrated, and when they are in this hydrated state, they have the perfect size to fit the sodium channel, and therefore they are allowed to freely diffuse through the channel in favor of the concentration gradient of sodium ions. On the other hand, potassium ions in their hydrated stage are way too big and they don't fit into the sodium channel, therefore being kept from freely diffusing through this channel. The selectivity of the potassium channel is also dictated by the inner diameter of the potassium channel. However, in this particular case, potassium ions, which are normally hydrated, are able to interact very closely with carbonyl groups that are present at the entry of the channel. This close interaction with those carbonyl groups leads to the dehydration of the potassium ions, therefore allowing the potassium ions to freely diffuse through the channel in favor of their concentration gradient. Sodium ions, on the other hand, are too small, and in their hydrated stage, when they try to enter the channel, there is too much space between the sodium ion and the carbonyl groups at the entry of the potassium channel, and this lack of enough proximity prevents those sodium ions from being dehydrated. Therefore, the sodium ions remain bound to the water molecule, and the combined size of the hydrated sodium ions is too large to penetrate the small diameter of the potassium channel, therefore preventing sodium ions from freely diffusing through potassium channels. At the molecular level, the potassium channel, the sodium channel, and the calcium channel exhibit a very similar molecular architecture. The potassium channel is made of a protein that contains 6 different transmembrane domains. This protein is repeated 4 times, therefore forming a channel that contains 24 transmembrane domains. The sodium channel is made of only 1 protein. However, this single protein is subdivided into 4 main domains, and each one of those domains is pretty much identical to the structure of the potassium channel. As you can clearly see here, this main structure contains 6 transmembrane domains that look almost identical to the potassium channel. This single structure is repeated 4 different times in the length of the sodium channel. Therefore, the sodium channel, just like the potassium channel, has 24 transmembrane domains. The same basic structure is also true for the calcium channel. In addition to the basic architecture of the channels being conserved among all of these channels, the actual function of the different transmembrane regions in each one of the domains has been pretty much conserved throughout evolution. The transmembrane regions 1 through 4 are the ones responsible for sensing changes in the electrical potential across the membrane, that is, in voltage. The transmembrane regions 5 and 6 are the ones that form the channel itself. And finally, transmembrane region number 4 contains several positively charged amino acids that are involved in sensing the electrical changes across the membrane, and these positive charges seem to be the ones that are responsible for the opening and the closing of the channel. Positive charges repel positive charges. Therefore, in order for the channel to be open, this particular transmembrane domain has to be displaced, has to be pushed aside so that the channel will be open. And speaking of the opening and closing of ion channels, let's talk about how the potassium and the sodium channels work in conjunction during the transmission of electric impulses, which, by the way, is the way we carry signals in the nervous system. And that is the process that we are about to look at is at the very root of everything that we do as humans. And the first concept that is essential to understand is that the concentration of sodium and potassium ions across the plasma membrane is not equal. Sodium ions are in very high concentration outside the cell, whereas potassium ions are in very high concentration inside the cell. This gradient of sodium and potassium ions is generated by the so-called sodium-potassium pump, shown here. This pump continuously brings potassium ions into the cell while moving sodium ions outside the cell, therefore moving both sodium and potassium ions against their concentration gradient across the membrane. And this is what defines this mode of transport of ions as active transport. The fact that these ions are being moved against their concentration gradient. Under normal conditions, the potassium channel is fairly leaky. That is, this looks like a faucet that can never be completely closed, therefore allowing water to drip out very slowly. The potassium channel is just like that. It allows the continuous movement of potassium ions across its central channel. However, this movement is very slow. Now, since the internal concentration of potassium ions, that is the concentration inside the cell, is substantially higher than the concentration of potassium ions outside the cell, the normal flow of potassium ions under resting conditions is from the inside of the cell to the outside of the cell. And this generates a resting potential across the plasma membrane. And that resting potential is about minus 60 millivolts in most neurons. The negative value of the resting potential indicates that the intracellular side of the plasma membrane is negatively charged compared to the extracellular side of the plasma membrane. When action potentials arrive, and by the way, action potentials are the same as a nerve impulse. When those signals arrive, there is a partial depolarization of the membrane down to a value of about minus 40 millivolts. When that value is reached, the sodium channel opens up suddenly, and the full opening of the sodium channel leads to the sudden influx of sodium ions into the cell, and this leads to a complete depolarization of the plasma membrane, followed by a repolarization of the plasma membrane up to a value of about plus 30 millivolts. That is, now the intracellular side of the membrane is positively charged compared to the extracellular side of the membrane. At this point, the voltage-gated sodium channels will close and the potassium channels will suddenly open. And the opening of the potassium channels, the full opening of the potassium channels, will then allow the flow of potassium ions from the inside of the cell to the outside of the cell in favor of their concentration gradient. And this flow of potassium ions outside of the cell will continue until when the voltage across the membrane reaches a value of about -75 millivolts. At that point, the potassium channels will close once again and they will remain closed for some period of time. It is important to mention that in the process of reaching the value of -75 millivolts, the potential across the membrane will actually cross the -40 range, which is the one that triggers the opening of the sodium channels. However, the sodium channel that is right in the direction of the incoming signal will at this point be in a refractory period. That is, this sodium channel will not be able to open for a certain amount of time. So when the -40 millivolts threshold is broken because of the flow of potassium ions across this open channel, this sodium channel will not be able to open, but another sodium channel located in this direction will be ready to be opened. And this produces the transmission in a unidirectional manner of the action potential. That is, the nerve impulse propagates in a unidirectional way along the membrane. In other words, the sequential opening and inactivation of ion channels is what provides directionality to the propagation of action potentials along the plasma membrane. And this is how neurons are able to propagate nerve impulses along their axons. Nerve impulses arrive to axons through dendrites that carry those impulses to the cell body and then to the axon. And those action potentials are then carried through the axon all the way to the terminal branches of the axon, where those signals are then transmitted to another neuron. And the transmission of those signals to the next neuron involves the action of ligand-gated ion channels. The best example of a ligand-gated channel is the nicotinic acetylcholine receptor. This channel is gated by the binding of acetylcholine to this pocket in the protein. Binding of acetylcholine opens up a channel that is permeable to sodium and potassium channels. The gate that is opened upon the binding of acetylcholine to the pocket is made of hydrophobic side chains. That is, there are hydrophobic amino acids in this region, and their hydrophobic side chains is the one that forms the gate that keeps ions from flowing freely across the channel. In close proximity to the gate of the channel, there are negatively charged amino acids, and those negatively charged amino acids are the ones that actually favor the flow of sodium and potassium ions through the channel. So the binding of the ligand to the receptor triggers a conformational change in this channel so that the gate opens up, therefore allowing the flow of sodium and potassium ions. The selectivity of this type of channel is determined by both the size of the channel and the charge of the area surrounding the gate of the channel. so that only ions of a certain size and a certain charge are allowed to go through the channel. And the combined action of voltage-gated channels and ligand-gated channels is what allows the propagation of nerve signals or action potentials from one neuron to the other. Therefore, you could argue that everything that makes us humans, our mental abilities, our intellect, our ability to think and act upon those thoughts, is all driven by the concerted action of ion channels. At the molecular level, that's what makes us be what we are, the opening and closing of ion channels. Finally, it is important to mention that what allows something as simple as the opening and closing of ion channels to allow for the complexity that is intrinsic to human behavior, human thinking, is the fact that a single neuron can be connected with many other neurons, hundreds of neurons, therefore forming very complex networks of neurons that communicate with many other neurons simultaneously. And it is the simultaneous activation of multiple neurons what leads to the formation of complex behaviors and what we consider to be human thoughts. This image, captured using confocal microscopy, shows you the body of a neuron together with its dendrites and the hundreds of presynaptic terminals represented by each one of those red dots. So the hundreds of presynaptic nerve terminals that are interacting with that single neuron. Each one of those presynaptic nerve terminals is likely to come from a different neuron. Therefore, the infinite complexity of neural networks. To finish this movie, let's take a very brief look at what happens at the end of those nerve terminals. In those regions of the neuron, nerve impulses trigger the opening of calcium channels. Once you have the opening of those channels, there is a substantial dramatic change in electric potential across the plasma membrane, and that leads to the fusion of these synaptic vesicles with the plasma membrane. And the fusion of these synaptic vesicles to the plasma membrane leads to the release of neurotransmitters to the outside of the cell. These neurotransmitters are then free to bind to the receptors that are located in the receiving neuron. And those receptors are ligand-gated ion channels. Therefore, upon binding of the neurotransmitter to the receptor will open up, therefore allowing the flow of ions into the cell. This flow of ions then generates a new electrical signal in the form of a change in membrane potential that will then propagate along the plasma membrane of the receiving neuron thanks to the action of voltage-gated ion channels. And this finishes another one of our movies. And remember, knowledge is